My name is Gabby Neal, and I'm going to be talking about the assembly of the primosome.
My name is James Shepard, and I'm going to talk to you about the process of initiation.
My name is Daniel Jezorensky, and I'm going to talk to you about the first steps of elongation.
My name is Olivia Bullard, and I will be talking about the last steps of elongation.
Here is our DNA replication model. The green strand represents the leading strand, while the blue represents the lagging strand. Now, the first thing that happens is DNA protein comes in and binds to the 5 9-base pair binding sequences, R1 through R5. Binding of DNA A causes melting of the DNA unwinding element DUE. Now, binding of protein DNA B, a helicase, to the single-stranded region of the lagging strand is helped by protein DNA C, where the helicase further unwinds the single-stranded region. This is now considered the pre-primosome. Then DNA C leaves the complex, and the helicase stimulates the binding of 3 primases, DNA G. Now the primosome is complete and ready to assemble the RNA primers.
The process of initiation is the assembly of the entire DNA polymerase III holoenzyme, consisting of a clamp loader complex and 3 core polymerases with associated beta clamps. The primases on DNA B will lay down primers on the leading and lagging strand in the direction of the replication fork. At this point, the lagging strand loops around itself to form the trombone loop, so that replication of the leading and lagging strands can proceed in the same direction towards the replication fork. This is facilitated by the single-strand DNA binding proteins. At this point, the clamp loader complex will open and close a beta clamp around the DNA near the RNA primers on both strands. The bound beta clamps now have a high affinity for the core polymerases. The tau subunits of the clamp loader bind the core polymerases and deliver them to the clamps. At this point, we are now ready to synthesize DNA.
At this point, the core polymerase begins synthesizing DNA from the primers in a 5' to 3' fashion from both the leading and lagging strands. The new DNA is represented by the orange yarn. The leading strand is synthesized continuously in a 5' to 3' direction towards the replication fork and must only be primed once. The lagging strand is replicated discontinuously in short pieces called Okazaki fragments. The trombone loop structure allows the Okazaki fragments to be synthesized in the direction of the replication fork. This is the way the replication goes.
The core polymerase on the lagging strand synthesizes from one primer to the next, then dissociates. Then, the DNA binding proteins on the next lagging strand fragment dissociates as well. Once the single-strand binding proteins have dissociated, a new trombone loop can form, allowing the core polymerase to bind and synthesize the next Okazaki fragment. This process continues until replication is terminated.